So howdy, I'm Chase Miller, and today I'm going to present to y'all how to do a Western blot. The first thing I want to do is present y'all with an outline, and that is that we're starting with what is a Western blot, and I'm going to move on to like why you want to do a Western blot, how knowing how to do a Western blot might benefit you in the future, and then I'm going to go into the procedural steps associated with a Western blot, which include sample preparation, protein separation, protein transfer, and protein identification. So the first thing is, what is a Western blot? A Western blot is a widely used analytical technique used to identify and quantify protein. And here I have an image of what a sample Western blot would look like once you finish. This is actually a Western blot used for identification of HIV in a patient. And the second thing is, why do a Western blot? How does knowing how to do a Western blot benefit you? So the Western blot provides useful information as to which proteins are present in the cell. And so this is applicable to primary research efforts as well as identification of disease in patients. The first step is sample preparation and involves extracting protein from the cell. So this is done by lysing the cell, and to lyse the cell you must introduce a lysing buffer as well as a protease inhibitor to the cell. The reason you're introducing the protease inhibitor is to keep proteases that are naturally in the cell from degrading the protein you're interested in looking at. So after adding the protease inhibitor and the lysis buffer, you're just going to pipette the cells up and down and then place them on ice for 30 minutes and add a couple extra reagents like BME and SDS. After lysing the cells, you're going to do a protein separation technique, which is most commonly done with an SDS page. The protein is loaded in a polyacrylamide gel, and then you're also going to add a sample, which is the molecular weight ladder, into the first well. After adding all of your samples to the polyacrylamide gel, you're going to put a current through the system, and because the proteins are going to be negatively charged, they're going to be attracted to the positive potential, and so that's going to cause the proteins to migrate, and they're going to migrate depending on their molecular weight. So that's how separation is achieved. After separating the proteins, it's important to transfer the proteins to another membrane. So this is going to be achieved with this wet transfer method. You're trying to transfer the protein from the polyacrylamide gel to either nitrocellulose or PVDF membrane. So to start this, you're going to have to remove the polyacrylamide gel from the cassette associated with the SDS page. And then you're going to place them in a wet transfer sandwich, is what they call it. And the steps are shown in the visual aid here. You start with 2 support grids, and then have pads and filter paper. And then your polyacrylamide gel is in the green, and your transfer membrane is like in between. And it's important when you're setting this up is that you have the polyacrylamide gel and the transfer membrane right next to each other, and the membrane should be closer to the anode than the cathode, because your proteins are negatively charged, and so they're going to be pulled to the positive potential again. And so if you had cathode, membrane, and then gel, then the proteins would be pulled in the opposite direction, and you wouldn't get any protein on your membrane. So the last step in the Western blot is protein identification. So now we've transferred protein to our membrane, and we're going to first do a blocking method where we add some protein, generally it's BSA, that interacts with the protein in the membrane. And that's to diminish non-specific interactions. So after blocking, we add a primary antibody, and this is an antibody specific for the protein of interest. And then we add a secondary antibody, and the secondary antibody is generally conjugated to something that will allow you to visualize this. It could be an enzyme, or it could be a fluorescent probe. But the secondary antibody is specific for your first antibody, so this is how you're going to get your output. And that's how you do a Western blot. Does anybody have any questions?
Yes. So when you did your Western blot, how does the first antibody know to bind to the second antibody?
So the second antibody, so generally you have a species-specific primary antibody. So your primary antibody might be derived from rabbit, and then your secondary antibody would come from another organism, like maybe goat, and it would respond to all rabbit antibodies.
Interesting.
How do you generate your primary antibody?
Primary antibodies are generated by, you can either have a hybridoma, which is a immune cell that's fused to a cancerous cell, so it's continuously proliferating, and then you would have had selected the initial immune cell from maybe a goat or a rabbit that had been inoculated with your protein of interest. All questions answered?
Yes.
Awesome. Thank you.